Yeah, so this is a presentation that I originally put together for the scientists at the Raleigh location. I showed you that slide where we were doing some causal model with sweet potato fries, and they thought it was interesting and wanted me to present about it to the rest of the research unit. And so, yeah, I think this is, you may not know what I'm talking about when I say causal modeling or structural equation modeling. I'll explain it and why it could be a useful technique in a lot of different fields of ag science. And I've used Calvin and Hobbes comics. Some of you might be too young for Calvin and Hobbes, but that was like my thing as a 90s kid growing up was Calvin and Hobbes. So I've tried to find some appropriate Calvin and Hobbes strips to illustrate my points. Okay, so, and I'll try to make this slightly interactive, so you'll have to answer a question or two as we go, but you won't have to do too much thinking. So let's get started. So, okay, fill in the blank of this famous saying. Correlation is not very nice. That's right. So, right, you practically already have memorized that as if it's some truth that's been handed down. Everybody knows that's true, right? You can't infer causation, one thing causing another, from a statistical association. That doesn't prove any causation, right? So yes, to some extent, that is true, right? It's kind of a good starting point, and you shouldn't automatically assume that one thing is causing another just because they have a statistical association. That's not wrong. But we're going to talk today about how maybe they can't prove causation, but we can infer and get some ideas about causation in complex systems just from data, from associations between the variables. Because you can read about Calvin's perspective on it, but the question that sort of we as scientists are trying to answer is why, right? We're not just feeding data into a box to get a prediction out. That is useful for some things, but we also are trying to learn and understand about the workings of the natural world. Like, we want to know why stuff is happening. So that's kind of the big question, right? And that's what we're sort of indirectly getting at when we measure things. But we want to really be able to be formal and precise about how we're answering that question. So first of all, maybe it's trivial to talk about causation because everything affects everything else. Like if some of one thing perturb the other even a little bit, they've had some effect on it, right? So, there's in physics, they say information or causal influences can travel as fast as the speed of light, basically. So there's, everything is affecting everything else. But maybe that's also too simplistic of a view. We can't say everything affects everything else, and we also can't pretend that correlation is not causation. So we just have to just shut our ears and pretend we can't say anything about causation. We just have to be careful and describe carefully how we think causes are working in our system. And by the way, if anyone has any questions, like raise your hand or just interrupt or anything. So that's kind of what I'm going to be talking about for the next half hour or so. And I want to begin by talking a little bit about the history of causal modeling. And this person I have on the screen is a scientist, a sort of geneticist, very famous from 100 years ago, Sewall Wright. He invented some fundamental statistical concepts like R squared. He basically was the first to use that, the F statistic for population genetics. He came up with a theory of genetic drift. I mean, he had very prolific, and his contributions are very fundamental to genetics, population genetics, and biology as a whole. So one of the things that Sewall Wright, one of the techniques he came up with is called path analysis, which is, he was interested in, if you're looking at a trait of a organism, what are the different influences of genetics and environment and their interaction on that trait, right? That's obviously the question in population genetics, right? But it's kind of a causal question, right? Like what, if you see a trait, what caused that trait? Was it genetics? Was it environment? Was it some of both? So he came up with this technique, path analysis, to kind of try to pull those things apart, is a causal model. But, you know, Sewell Wright kind of got overshadowed a little bit by this dude. He's like the villain of our story, R.A. Fisher. I mean, he basically single-handedly invented statistics as we know it. But, you know, he also, if he had beef with you, he let you know it. Like if he didn't really think that it was right to say anything about correlation, about causation from correlation, but he didn't just say, hey, I respectfully disagree with your paradigm. He said that's completely wrong, and just like work to tear down that idea and make it so that no one would ever try to infer causation from correlation ever again, basically. And that kind of worked for 60, 70 years. It was sort of the path analysis thing was kind of a dead end. No one really developed it anymore because they were also afraid of like making this guy Fisher mad. I showed a picture of him smoking tobacco because he also later in life kind of like was a shill for the tobacco industry and used this idea of, hey, no causation from correlation to help sow doubt about the links between tobacco use and cancer and other diseases. So, you know, obviously there's kind of like a dark side to this, I think, right? Like obviously, yes, it'd be great if we could have a randomized controlled experiment for everything to just say once and for all, do cigarettes cause cancer or whatever? But if you can't do that, you can't just say, well, then I guess we have to give up and say we don't know. There are other ways to get to that relationship besides doing an experiment on everything. So luckily, we've kind of moved past that old debate, I think, a little bit. And so, yeah, like I said, Fisher's influence lasted for generations, but it started to break down a little bit in the last couple decades, especially this man here is one of the key reasons for that. Judea Pearl, he is a pioneer of causal modeling and the use of DAGs, directed acyclic graphs, which we're going to talk about what that means to infer causation in complex systems from data. He wrote this cool book called The Book of Why, kind of showing his, he wrote lots of academic papers, but that's kind of his more popular book on the subject. So we're kind of saying, okay, let's make up between this old debate, and yes, we can't infer causation from correlation, but we can get pretty darn close. So now we've got the history, and that's cool, but let's talk a little bit about causal analysis. So let's say we want to know what is the relationship between an independent variable, a predictor variable, X, usually I call it X, and a dependent variable, response variable, usually call it Y. Now, these variables might be related. There might be a correlation between them, but those aren't the only variables in the world. There's other things in the system that are going on. And there's many ways in which the relationship between these two variables can be influenced by a third variable. And let's talk about that. So the first way is, you may have heard the term confounding variable or confounder. What does that mean? And we'll talk about it in a minute. And so these graphs help to illustrate what's going on. We have confounders, mediators, and extraneous variables. So anytime an arrow is pointing from one variable to another, that means there's a influence of that variable on the other. There's a cause operating. So a confounder is a variable that affects both the independent and the dependent variable. You see the arrow going to both. I'll give examples of all these in a minute. Mediator, a mediating variable, is one that The independent variable affects the mediator, and the mediator affects the dependent. And an extraneous variable is one that has its own influence on the dependent variable, but is not related to, there's no path going that involves the independent variable. It's a separate influence on the dependent variable. So you can see these are basically graphs, network graphs showing the relationship between variables. And so there's sort of three different ways that you could draw arrows between three variables, and this is them. So let's go with some concrete examples. So let's talk about confounder. Okay, let's say we measure, this is kind of the classic example. Let's say our variable is ice cream sales. How many ice cream cones are sold in some location, like a beach? And the dependent variable is how many, at a certain time point, how many shark attacks there were at that beach. So we've measured these two variables. We calculate a correlation. We find that they are strongly positively correlated. So what would be a possible confounding variable that could be confounding and affecting both those variables? So some people say number of people, time. Yeah, so it could be a per capita thing where the more people, the more ice cream, and the more people, the more sharks. Another one that I was thinking of is temperature. Like if you look at it by season, more people went to the beach in summer, there was more ice cream sold, but also more people went swimming. So if you see that relationship over time, maybe it's just due to the season of the year. Now, in this case, right, there's really no causal relationship between ice cream and sharks. Sharks are not attracted to ice cream or whatever. So if you statistically accounted for that confounder, the confounding is so strong that there would be the remaining effect that would be left would be pretty much zero, right? This is a very, very strong confounder, but not all confounders are quite that strong. You can also have just like partial confounding effects. But that's sort of the classic example of a confounder. Now, a mediator, as I said, is one that there's a chain of influence. Independent variable affects the mediator, and that influences the dependent variable. Okay, here's another example. Let's say we measure some students' grades and their happiness and find that there is a strong positive correlation between their grades in school and their happiness. What would be a possible mediating variable between the two? Sorry? An award? Maybe so. Anyone else? Okay, maybe. Yeah, so there's, right, grades maybe don't directly affect happiness. You could say they do, but you could also argue that it operates through some specific mechanism. And like maybe you win awards and those are really what makes you happy, not the grades. That's a possibility. So, but you could also say like, well, maybe grades are affecting self-esteem or some other variable, which then people with high positive self-image tend to be happier or whatever. You know, you could come up with mediators for lots of things like that. That's just one example. And the extraneous variables are ones that influence the dependent. They have an influence on Y, but not involving the independent variables in our model. So now if we have extraneous influences in a statistical model, that's like noise. Those are what we call the residuals of the model, things that we just didn't account for. So it's not necessarily essential for you to account for these in your statistical model because they'll just come out in the noise. But the more extraneous variables you capture, the better you can explain the thing you care about. So it's good to measure these things and account for them. So here's an example. Let's say we measure the blood alcohol level of drivers and then their distance that it took them to stop their car, the braking distance. What might be an extraneous variable that also affects braking distance, but doesn't have anything to do with alcohol level? Right. So there's obviously like lots of things that affect braking distance that have nothing to do with the reaction time conditions on how much alcohol you drank, like weather and that kind of thing. So that would be if you did an experiment where you measured that, but there was different weather conditions you didn't account for in the model, that would be like in the noise. It would cause variation that you didn't explain. So you want to minimize these and control for them as much as possible, but they're not essential. Like it would be, the confounder really can mess things up and give you the wrong inference. But these are just kind of noise. So all those little 3 circles with arrows connecting are called DAGs that I showed you so far. What is DAG? What does that even mean? DAG stands for directed acyclic graph. But you probably don't even know what directed, acyclic, or graph even means, so that doesn't help you too much. Well, the directed part refers to the fact that the different variables are not just connected by lines that could go either way, they're connected by arrows that have a direction to them. That shows we're saying there's a direction of causal influence going from one to another. Like blood alcohol level affects braking distance, but the braking distance of your car doesn't go back and affect how much alcohol is in your bloodstream, right? It only goes one way. Acyclic means there's no loops. The way those arrows are drawn, you can't make a loop around and get back to where you started. If you start at variable A, the way those graphs were, you can't go back to A. There's no cycle, there's no loops in it. The influence only propagates through in one direction. And a graph is not like the colloquial term, but it's a mathematical term. A graph is a mathematical structure which is pairwise relationships between a set of objects. So those different variables are like nodes in the graph that have relation between them. That's what a graph is. If you're a mathematician, which I'm not, if you say graph, they don't think, oh, like a scatter plot. They think of variables connected by relationships. So that's what a DAG is. And I've shown you those simple DAGs were the 3 variable relationships, the mediators and confounders and that kind of thing. But pretty much all DAGs could be built up, even if you put more variables in, out of those building blocks. Those little structures of confounders and mediators can be put together like, tinker toys to make more complicated relationships if your system has more than 3 things going on. So that was kind of the background to get into a little more of how do we translate all that stuff I just talked about verbally into a statistical model with actual numbers on it that you could say something about or like publish in a journal or whatever. Well, we can use a technique called structural equation model. We can take a DAG, draw it however we want to on a whiteboard with circles and arrows, and then translate those arrows and circles into a system of equations that show the relationships between 2 or more variables. So it's like taking one of those graphs and writing it down as a bunch of equations showing the relationships. That's what a structural equation model is. SEM, people call it. And so SEM is not just, people in here have probably done multiple regression where you have a response of a bunch of different variables that are trying to predict that response. In a multiple regression, you have a Y variable and a bunch of X variables. A variable can either be on the right-hand side or the left-hand side. It can't be both. It can't be an X and a Y. But the cool thing about the structural equation models that make them better able to capture what's going on in the real world is a variable in there can either be an X or a Y. In one, it can be an X in one equation and a Y in another. So it's different than just a regression with a lot of variables. So like one variable can cause another, and then it can cause another variable. So it can be X and Y. So I just mentioned this, but I'll show you graphically how does a structural equation model's graph compare to a simpler regression model that you're probably familiar with. So, right, this is even a simple linear regression has a DAG, right? It's just X causes Y. That's it. There's one equation there. That's a simple linear regression, right? Multiple regression would look like this. All the x's are affecting y, but y doesn't affect anything else, and the x's don't affect each other. They all independently affect y. Even if you have interactions, that's just like an extra variable that comes in and affects y as well. But this is like what you could make something like this. This is just one example of an SEM model you could make with 3 predictors and a response. Like x1 affects x2, which affects x3, you know, but not x3 doesn't affect x2, for example. Like that's just one example. The possibilities are a lot more when you talk about SEMs. And just to throw you a little bit of jargon in SEMs, they talk about a variable is either endogenous or exogenous. And an endogenous variable is, at least in part, explained by other variables in the model. So, okay, if this is my DAG, what variables are endogenous variables in this model? Anyone have a guess? X3, yep. Any other ones? There's one other one. Y, yep. Anything that has at least one arrow going into it, endogenous variable. There is some other variable in the model that's explaining at least part of that variable. And all the other ones, so right, x1 and x2 have nothing going into them. There's no arrow going in. So those are exogenous. They originate outside, but they have residual. All the variables will have residuals, and there might be covariance between them, but that's just kind of part of the noise, essentially. Right, so those are, you were right to identify those as endogenous and those as exogenous. And then we have, so right, you might say, well, we also want to estimate the covariance between the other ones that's not part of this graph. So now it's not a directed acyclic graph anymore because you have a two-way path. It's not all one way anymore. But that's okay. We can still estimate those correlations with our structural equation model. So we're still good. So now I mentioned path analysis when I started talking about Sewell-Wright a while ago. But path analysis is something that you can do once you have estimated the parameters of a structural equation model. You can use path analysis to look at overall direct and indirect effects of some variables on each other. So let's say we want to what we really care about in our science question is the effect of A on B. Let's say that's like what we really want to know. Well, we estimate all these parameters to like look at they're basically the individual relationships between the different variables. And you can see that there's different paths going from A to B, right? One of them is just a straight path going straight from A to B. One of them goes from A, C, C to D, D to B. One goes A to E and then E to B, right? There's 3 different ways. If you start at A, 3 different ways you could get to B. And any one of those represents some effect that A has on B. A has a direct effect on B, and it has some indirect effect because A affects C, C affects D, and then D affects B. The way you would add up path analysis is basically multiplying and adding up the different parameters to figure out those overall effects. So like the product, if you want to know how A affects C, C affects D, and D affects B, you would multiply the effects up. So like and as you know from basic math, arithmetic, a positive number times a positive number is positive, negative times negative is positive, positive times negative is negative. So like if A has a positive effect on E, but E has a negative effect on B, then if A goes up, E goes up, but B is going to go down. So A is having a negative effect on B because A is making E go up and E is making B get less. So A has a negative effect overall, right? And if it was 2 negatives, if A makes E go down, and then if E goes down, B goes up, the 2 negatives multiply to a positive, and that means A is actually having a positive effect on B, even though they're both negative. And then, so you multiply paths up within the path, and then you add them together if you're adding multiple paths together. So the net effect on each path is multiplying these. The total effect is you sum up all the direct and indirect paths to get the total effect of one variable on another. I'll give a concrete example in just a second if that kind of went over your heads a bit. So like if you did it in terms of actually multiplying the parameters, this is what would be the total effect of A on B. You'd have this beta AB is just this one direct path, and then the other are the product, AC, CD, DB, and then the AE effect and the EB effect. That's how you would calculate it if you're looking at the causal relationship. Okay, so here's a simple example with only 3 variables just to make it a little easier for you. Let's say we did a study where we want to know, does exercise make you lose weight, okay? And we find that these are, after we collect all our data and estimate the parameters of the structural equation model, this is what we get. We get positive 0.3, positive 0.15, and negative 0.44 are our, as we also look at food intake, right? So if you eat more food, does that cause you to gain or lose weight? Now, what is the direct effect of exercise on weight loss? Which number in here? 0.3, right? So there's a positive direct effect of exercise on weight loss. Now, the indirect effect on weight loss via food intake, how would we calculate that? Close. It's 0.15 times minus negative 0.44. You would have to multiply the two numbers together because, right, if you exercise, it makes you want to eat more, which then mitigates the effect of weight loss because you're eating more. So there's a negative indirect effect. And how would you calculate the total overall effect? What would you add up? That's right. You would take 0.3, subtract 0.07. So we still have a positive effect. Sort of exercise promotes weight loss, but because exercise makes you want to eat more, it kind of mitigates the effect a little bit, and the overall effect is not quite as strong as you would predict if exercise didn't impact food intake at all. That's the kind of thing. Those are the kind of questions you can answer with path analysis, with structural equation models. Obviously, that one's hella simple, but you can do much more complicated ones than that. And this is just to give you a little taste of what you can do. So I wanted to also just mention, since we're all kind of ag scientists in here, are people using this in ag science? Well, it is pretty common technique in the field of ecology because ecologists are looking at complex systems with a lot of, organisms interact with each other and affect each other's biomass in a system, for example. And so I would say in ag science, the main place so far that I've seen these things used is agroecology. And here's a paper I found that just came out, last year, where they looked at species richness of bees on like a sunflower crop and looked at whether or not, some management treatment was affecting that, and by what mechanism does the management treatment affect the bee richness? Well, it could be because it's affecting how many different species of flowers are there, or how the biomass, or like the amount of the flowers, and you can kind of tease that apart with the path analysis. So this figure was it's a DAG basically with the model parameters on it that were estimated from one of these models we're talking about. And yeah, well, since I first gave this talk to a bunch of food scientists, I was trying to sell them on the idea that your food chemistry is a complex system with a lot of variables, and the SEM approach is ideal for that, for these questions where it's okay, we want to know, I think so yeah, in the sweet potato fry world, hardness, smoothness, oiliness, and roughness, which is taste attributes of the fries, are super important, and they want to have them just right so that more people will buy the product. But it's like, well, what affects that? Is it the, you know, the amount of starch that's there? And then I don't have no idea what all these things are, but they're like chemical properties that you can measure which one of them is actually right. Like if you want to like breed sweet potatoes to make more, I don't know if oily is good or bad, but like let's say it's good. If you want to breed sweet potatoes to make more oily, you could actually like figure out what's really causing that and breed more precisely if you know the true, not just the correlation, but really what is causing it. Then you actually can do a better job breeding for that thing you care about because you can manipulate the actual thing that's causing it, right? Like it'd be good to identify what part of the chemistry is actually causing the things we're can use to make money off the sweet potato fries. So that's an example there. And another thing that SEMs can be used for, yeah, you don't have to care exactly about this figure, but there's this idea of latent variables where like you can, in food science, right, they give tasters some kind of like questionnaire to fill out of like different attributes. And those are just the whatever they write on that test is the variable, that's your data, but it's actually only indirectly estimating sort of an underlying variable of like whether or not their actual preference for the food. And so you can actually put variables that aren't even observed in your structural equation model. If you think that like sort of this overall sugar variable or overall flavor is what's influencing what the tasters are saying about different, like they rate things on a scale of 1 to 5, you can actually model that overall flavor variable that you can't ever truly measure, but you can put it in your graph and actually estimate it. So that's kind of a cool thing. And my opinion is, and this is what I was kind of like trying to sell when I first gave this talk, is that SEM, this technique is really woefully underappreciated and underused in ag science, and we should be using it a lot more. Because a lot of times what I see when I work with ARS scientists' data is people do a study. It might be an experiment, it might be observational, but you know, they have some experimental treatments or some groups they're observing. A lot of they measure a lot of response variables that may or may not be related to each other. So you have like a couple treatments, and then you measured like 10 different variables of response. Well,
real quick, so I actually got to pick up Rye from school.
Can you please mute yourself? I'm sorry. Thank you. See if I can. Let me see if I can mute the. There we go. All right. Yeah. So what they do is they have a couple treatments. They measure like 10 variables, and then you do an ANOVA for the effective treatment on biomass, an ANOVA for the effective treatment on, you know, whatever, like protein content, and ANOVA for the effective treatment on oil content. They just like, then your paper is just like a stack of ANOVAs that you publish. And that's great. Like that is good science and that's useful, but you're missing out on maybe the interrelation between those different variables. Those aren't just all independent. They're affecting each other. And if you don't, you could be missing some interesting stuff that's going on that could actually give you insight into really what you should be breeding for if things are affecting each other. You know, you could be missing an insight that will allow us to learn more or allow us to make better crops or whatever your goal is. So I think that we should be using this technique more than we do right now. Yeah. So yeah, like I said, we're really missing out when we just throw in a pile of ANOVAs. But before I get you all sold on this, let's give a few caveats of maybe some backs or just things to keep in mind when you're doing this kind of stuff. Now, this isn't some magic, right? Like this is just another linear regression model, just like an ANOVA or anything else. Like it makes a lot of assumptions, just like those ANOVA models do. And right, like a linear model assumes the relationships between variables are linear. Like that's kind of the key assumption they make. If that's not true, a linear model is not good. So you had to just keep that in mind. And, you know, obviously normally distributed error is a big thing in linear models. We can relax that assumption in various ways, but by default, that's what we assume. And so you have to make sure that assumption is valid, even if you're doing SEM, just like you would look at the residuals of an ANOVA to see if they're normally distributed. You still have to do all that same stuff. You don't get to like have a free pass on any of that stuff. And another, well, this is an especially important point in SEM world. You should always standardize your variables. If you're doing any type of multivariate analysis, including SEM, variables are measured in different units, right? So like you might have one that's micrograms per milliliter and one that's kilograms, or they're all, different orders of magnitude and everything. They're not standardized. So if you have a statistical model that has all these different variables in it, the parameter size of the parameters is going to depend on the unit. So you just have to make sure you always know what you're comparing and either know what units are in, or you could just z-transform everything or standardize it to make sure you're comparing what you think you are. And I just said this already, but SEM is not magic. It kind of depends, just like anything, it depends on the question you ask. So we're trying to, Calvin is trying to figure out causal relationship between TV watching and violence, and is just like R.A. Fisher is exploiting the doubt that like, well, we don't have a true proof that TV causes violence, so we can avoid having to worry about it. But like I said, there's no magic solution to that problem. SEM can compare different causal hypotheses to each other. It can't say for sure that there's causation, because it's still true that you can't really prove causation from correlation, but you can compare different hypotheses about causality to each other and say which ones are the more supported by the data, right? So just like anything, we have different hypotheses and we're saying which one is more consistent with the data. And just like Calvin says, the trick is asking the right question, which means don't just throw your data in and see what happens. You should have a theory behind what you're doing. You're all experts in your field of whatever applied science you work in. So you should have some ideas of how you think that system will work and build your models accordingly. You don't just let the computer decide, And another thing that SEM should not be used for is fishing for statistical significance in the data. Like, it shouldn't be used to just blindly throw in all the variables and see what's significant, because it's kind of hard to do when you have a model that has all those complex different ways that things could be related. Think about all the permutations you could make. I mean, you can't really just blindly try out every possible graph when you even have like 10 variables. There's like billions of graphs you could make, so you can't test them all out. What you should do is use what you know and your theory that's behind it to construct a small number of well thought out hypotheses. One hypothesis being a graph of how the variables are related. Make a small number of them and test them against each other. And a good way to do that is like just getting on your pencil and paper or your whiteboard or whatever and draw those boxes and arrows yourself by hand, and that'll help you visualize how things are related. And then once you have your ones that work, that you like, you can translate those boxes and arrows into equations, into a statistical model, and see which one the data support the most. And just as I finish this talk here, I'm about done, but I just want to throw out there that I've just given you the very superficial idea of this. There's lots of books about it. There's ways to do nonlinear relationships in a structural equation model. There's ways to do non-normal errors if that's what you're into. You can do anything you could do with a regular statistical model. You can do with the SEM model. You can do random effect too, which is nice for our agricultural experiments like block design and agronomic studies that are done at multiple locations and that kind of thing. But like I said, SEM is still in a phase of development. So all these techniques are kind of still being actively developed by statisticians and by applied scientists. So if you ask again in a few years, you could have a different answer to like what you can do with these type of models. And I've been hitting this point a few times that there's kind of, people think of, most people, at least in ARS, are experimental scientists. We do manipulative experiments. We don't usually go out and just collect observational data without manipulating something first. And I think it's thought of that these kind of causal models are more for observational data. If you have an experiment, you can just like, your treatments are supposed to be the only things that are affecting the response. So why do you need this causal model? We just model how does the treatment affect the response. So some people say that, but even the most well-designed experiment is going to have covariates. It's going to have interactions between the different things the responses you're measuring are affecting each other. Even if your treatments are perfectly controlled, the different responses you measure affect each other, and they could confound each other. And you can use an SEM to help you pick that out. And yeah, I'm just going to end by saying that there's a bunch of R packages, and SAS has some procedures for doing this kind of modeling. So these are the ones you should look into if you think this is cool and want to try it out on your own data. There's also, this nice website called Daggity that you can use to draw the actual graphs and make nice figures of them. And it will do network analysis basically on those graphs that you draw. And well, you already know the SEA stats page, so you don't need the QR code, but that's all I have on causal models. So I'm happy to take any questions.